Good evening. As this is the first episode of my new channel, I wanted to make sure it was positive, and this report from the United Kingdom I think certainly qualifies. Almost everyone has a story about losing a loved one to cancer. This news report is personal to me as I too have lost both family and friends to cancer. But I'm happy to report that scientists may have accidentally found a powerful tool or even a cure in the fight against different cancers. Before we continue, please hit that like button and consider subscribing to the channel. You can always unsubscribe if my content is not up to your standards. Throughout history, mankind has stumbled by accident onto some huge discoveries that changed the world. Here are a few examples. While working on radar, microwaves were discovered by accident. A German physicist was working on a cathode ray tube, like the kind used in old televisions, when he accidentally discovered X-rays. While studying lysogenic acid, a chemical derived from fungus, a chemist accidentally created and ingested LSD. While working on heart medication at Pfizer, the resulting drug was ineffective against heart disease, but it had other reactions. The drug was rebranded, and Viagra was born. While working at DuPont, a chemist was researching refrigerants, the kind that go in fridges and air conditioners, when by accident he created something that would be almost magical in the world of cooking: Teflon. If the results of this and further studies hold true, this accidental discovery of beating cancer could be right up there with those huge discoveries of the past. A British newspaper reports, researchers at Cardiff University were analysing blood from a bank in Wales, looking for immune cells that could fight bacteria, when they found an entirely new type of T-cell. That new immune cell carries a never-before-seen receptor which acts like a grappling hook, latching onto most human cancers while ignoring healthy cells. What makes this new discovery so exciting is that prior therapies, which use immune cells to attach to HLA molecules on cancer cells' surface, fight cancer but are incapable of fighting solid tumors. HLA molecules vary in people, but the new therapy attaches to a molecule called MR1 and not HLA, which does not vary in humans. This gives it the chance of fighting most cancers, and additionally means people could share the treatment, allowing banks of the cells to be stored and thus be offered quickly to people suffering from the disease. What is absolutely astonishing about the new treatment is immune cells from the new treatment have killed lung, skin, blood, colon, breast, bone, prostate, ovarian, kidney and cervical cancer cells. That's absolutely fantastic. Professor Andrew Sewell, lead author of the study, said, "This was a fantastic finding. Nobody knew the cell existed. Our finding raises the prospect of a one-size-fits-all cancer treatment, a single type of T-cell that could be capable of destroying many different types of cancers across the population. Previously, nobody believed this could be possible." Astonishingly, T-cells of cancer skin patients, altered by the treatment, were apparently capable of killing the patient's cancer cells and other patients' cancer cells as well. The team of researchers said that trials on terminally ill patients could conceivably start in November. Professor Oliver Ottman, Cardiff University's head of hematology, stated, "This new type of T-cell therapy has enormous potential to overcome current limitations of CAR-T, which is a different kind of therapy, which has been struggling to identify suitable and safe targets for more than a few cancer types." Professor Arwin Gilmore, I hope I'm not butchering the name, of Cardiff University's Division of Infection and Immunity and Cancer Immunology, lead for the Wales Research Centre, added, "If this transformative new finding holds up, it will lay the foundation for a universal T-cell medicine, mitigating against a tremendous cost associated with the identification, generation and manufacture of personalized T cells. This is truly an exciting and potentially a great step forward for the accessibility of cancer immunotherapy.